Good afternoon, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
My name is Bunga Laili. It's a precious chance for me to be your master of ceremony on this very special occasion. The fish stock in pandemic area, ornamental fish and heat shock protein. First of all, let's say thanks to God who has given us guidance, happiness, healthy, and mercy so we can participate in this special event without any obstacles. On this special afternoon, we have several agendas as follows. First one is speech from the head of Research Institute for Ornamental Fish Culture, Dr. Idil Ardi. Second, main lecture which will be delivered by three keynote speakers. They are Ahmad Musa, Master of Science, who will present Indonesian ornamental fish future research challenge and opportunities. Dr. Charles Saliba, who will present the role of heat shock protein and journey into HSP stimulant finding. And Marta Arendodo Luque, Master of Science Aqua, who will present application of HSP stimulant in aquaculture industry. Well, ladies and gentlemen, let's begin this event by reciting Bismillahirrahmanirrahim. Now, I would like to invite Dr. Idil Ardi as a head of Research Institute for Ornamental Fish Culture to give a speech. Dr. Idil, please, time is yours.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, praise be to Allah Ta'ala, who has given us knowledge, which when compared with his knowledge is no more than a drop of water in the ocean. Our blessing and greeting are sent to the great Prophet Muhammad, peace be upon him. Our respect to speakers, research professor, researcher, lecturer, academic, and fisheries stakeholder have presented in webinar. I present here to open web conference or webinar today to represent our head of Research Center for Fisheries, because at the same time, he is incidentally invited by head of Research Agency and Human Resource of Ministry of Marine Affairs to meeting. In this time, I say his apologies to all of us, especially to speaker Mr. Charles, Mrs. Marta, and Mr. Ahmad Musa. Well, I want give about information about ornamental fish in Indonesia. Indonesian ornamental fish consists of various types, both endemic and unique. Almost all major islands have their own endemic fish. There was super red arowana in Kalimantan, botia and tiger fish in Kalimantan and Sumatra, banggai cardinal fish and rainbow fish in Sulawesi, rainbow fish in Papua, as well as several rasbora species on the island of Java. All of these species could become extinct if there were no cultivation tools and catch control. Ornamental fish research, especially for aquaculture, is still lacking priority. Apart from the fact that ornamental fish commodities are not yet a top priority. but also because it is realized that the primary need of our society are fish to meet for food needs. But this does not mean that we will leave the research of ornamental fish, no progress or no development. This why we have this research institute for ornamental fish culture as a message to the world that we serious about maintaining our separate in the field on the ornamental fish. Ladies and gentlemen, today's program is called Fish with if translate into Indonesian language maybe more accurately regarded as bincang-bincang. The topic presented began with the title Indonesian Ornamental Fish Future Research. Challenge and opportunities will be presented by Ahmad Musa from the Research Institute for Ornamental Fish Culture in Indonesia. We hope that what will be presented can become material for future research development, especially Indonesian ornamental fish. We will also invite Dr. Charles Saliba from Nutribiotic and Malta to bring the presentation entitled The Role of Heat Shock Protein and Journey into the Heat Shock Protein Stimulant Feeding. We hope that what will be presented can be a motivation for researcher and fishery stakeholder as well as scientists in other fields. How to continue research into something that can be commercialized. At the end of the event, there will be presentation by Marta Arredondo, Masters in Aquaculture from Palmer, Technofit Spain, with the application of heat shock protein stimulant in aquaculture industry as an illustration of how a product produced can be applied and tested in the field. Ladies and gentlemen, what is a correlation between fish, especially ornamental fish, with heat shock protein? We will listen carefully from the exposure of the speaker, even though term heat shock protein is something that has been very busy being discussed among scientists. But maybe some of us are still less concerned about the role in the physiology system. Hopeful with this event, it can add insight to audience as well and can be follow up with more concerned action. Our hope have nice discussion, especially for Dr. Charles Saliba and Marta Arredondo Laqui, Master of Aquaculture. We can thank you for participating in this discussion. We also do not forget to convey our best regard to Dr. Kartik Barwa from Sheffield Lemberg Universities. With this speed, a very long journey and has become an intermediate, so this discuss could be conducted. Finally, I say thank you very much to Pak Musa or Mr. Musa and team have prepared this webinar, web conference perfectly. I think so. I say thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Dr. Idil Ardi. Before we start the seminar, we would like to invite all of the participants to join us for the photo sessions. Please turn on your camera to let us capture your screen. Okay. Smile, ladies and gentlemen, you're on camera. Let's do.
Sekali lagi.
Once more. Okay, once more. Okay, the photo session is done. Thank you for all of you. After this, the seminar will be led by our moderator, Dr. Ofri Johan. To Dr. Ofri, please, time is yours.
Thank you, Mbak Bunga. Ladies and gentlemen, the international webinar with the topic Fish Stock in the Pandemic Era, Ornamental Fish and Heat Shock Protein is going to begin. Good afternoon for participants in Indonesia and good morning in Europe time, because two speakers today from Europe. Allow me to say in Arabic, Assalamualaikum warahmatullahi wabarakatuh. Honorable for Dr. Idil Ardi, Master of Science, Head of the Research Institute for Ornamental Fish Culture. Distinguished guests for three speakers, Ahmad Musa, Master of Science from Research Institute for Ornamental Fish Culture from Depok, Indonesia. Second speaker, Dr. Charles Saliba, Nutribiotic Ltd from Republic of Malta. The second speaker, Marta Aridondo Luki, Master of Science, Aqua, Farm Fee Technofit from Spain. Introduce myself, I'm Ofri Johan. I will be moderator in this webinar. Ladies and gentlemen, I hope everyone having a good time. First of all, I would like to welcome you all to join this talk. The presentation will carry out about 20 until 30 minutes for each speaker. After all speakers gave presentation, we have a time to discuss by asking a question about 15 minutes. I'd like to choose participants who gave a sign and who write on their question in the chat room, whether in Zoom or YouTube. After question and answer, I invite Dr. Kartik Berua. I hope he will be join us. And let's start. In this occasion, we would like to have a presentation from Ahmad Musa, Master of Science. Before the presentation begin, allow us to introduce the presenter. Ahmad Musa was born in Makassar, 28 February 1980. He graduated from Chemistry Department of Hasanuddin University in South Sulawesi, Indonesia, and also Magister of Aquaculture from Bogor Agricultural University, also in Indonesia. He is a researcher in Research Institute for Ornamental Fish Culture since 2006. Various research already joined by Ahmad Musa from the field survey till laboratory experiment. His main work is related to protein purification using electroelution after SDS page, hormonal analysis using ELISA, Western blot, developing ELISA assay, rising polyclonal antibody, agarose precipitation test, coagulation test, and human research deployment. Some publication and 2 national patent has been submitted until today. Today, the presentation title we will give Indonesian ornamental fish future research and challenge and opportunities. For the participant, just short information. After 3 presentations, there will be 2 sessions, question and answer, and each session is for 2 questions of each speaker. Don't forget to mention your name, affiliation, and country, as well as for whom the question is. Without further ado, I would like to welcome the presenter to deliver presentation. Mr. Ahmad Musa, the floor is yours.
Thank you, Dr. Ofri, for the opportunity. Allow me to give share screen. Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Dr. Ofri Johan, for the opportunity. With respect, Head of Research Institute for Ornamental Fish Culture, Dr. Idil Ardi, Dr. Charles Saliba from Nutribiotech, Malta, Mbak Marta Arredondo Luque from FAMPAI Technopit, Spain, and also I have Dr. Kartik Barua already present also from Sveriges Lantbruk University, Sweden. My senior research professor, researcher, lecturer, student, and fishery stakeholders, please allow me to deliver simple presentation with the title Indonesian Ornamental Fish Future Research Challenge and Opportunity. This presentation will not telling about how to cultivate the ornamental fish, but more like pieces of idea during the activity in ornamental fish research since 14 years ago. This is a happy day because I show you in the screen, this is a first letter for me. It's in 23rd June 2006. It was the first sign for me as an officer in this institute. So it's 14 years ago. So actually, I hope this event could be conducted yesterday, but because lack of time, so we conduct it today. Before going far, we discuss about the ornamental fish. What is ornamental fish? Many define the ornamental fish, but Simply, it could be said that ornamental fish are fish which rear alive to be enjoyed visually. Different from the consumed fish, however expensive an ornamental fish is, its price will fall and even worthless if the fish are dead. There are at least 3 categories that cause fish to be included in the ornamental fish category based on unique, rare, and subjectivity. When a species is considered as a rare species, many will be interested in collecting to maintain it. For example, ancient fish or living fossil, Latimeria menadoensis. I'm sure there are many people who want to collect this fish even dead, moreover alive. Based on its uniqueness, ornamental fish could be considered unique not only because its color, but also because of its shape and behavior. Rainbow parrot is a unique example. This is a unique example of ornamental fish, and also archer fish or Toxotes jaculatrix is considered unique because of its behavior, which shoots its prey with the bars of water from its mouth. Also, as an example here, laka fish from the Gobiidae family in the La Susua River. The unique characteristic is that the abdominal fins are united into discs. Local resident identify as laka or a tage. The disc cause this fish could stick to the rock surface when resisting swift currents. These fish are often found attached to rock surface, even on the barrel wall, as I show you in the picture. As for the subjectivity factor, the trend commodity or hearsay factor as good-looking fish, even a person's interest could automatically lead to a species being considered as an ornamental fish. So don't be surprised if one day you see someone who maintain eel as an ornamental fish because it's an interest matter. Based on commercial factor, there are at least 3 category or 3 level of ornamental fish: local, national or regional, and international. Fish found in ornamental fish market at least could be categorized as a local ornamental fish, and when we find fish that is transported between islands, it will at least be recorded in the fish quarantine agency and could be considered as a national ornamental fish. To be categorized as an international ornamental fish, of course, at least data from exporter need to be obtained. Regarding its benefit, at least this ornamental fish provide benefit from economic, health, science, psychological, and environmental aspect. I would like also to talk a little bit about our office, the Research Institute for Ornamental Fish Culture, which is the only government agency in Indonesia that has the main task to conduct ornamental fish research. It established in 1957 for freshwater fish, but in 2002, its main task became ornamental fish. Many success stories have been produced, one of which is as the first in the world to breed botia or Chromobotia macracanthus outside their habitat in a controlled system and continues until today. I will not convey this success, but rather want to emphasize the various obstacles and problems we face in doing research so far. This kind of the fish that we maintain here, both endemic, native, and introduction. This is what I mentioned before. This is the fish that we are the first in the world to breed this outside in the habitat. And a lot of fish. What are the problems that we face in ornamental fish? When we go to the natural habitat, the first problem we face is the public awareness. Some of them are, we often facing this matter in an expedition for collecting fish from natural habitat. We found a decrease in the number of fish that could be collected, and after searching, we found that it was caused by human error factor. The human that didn't pay attention to the sustainability of an ornamental fish. For example, Banggai cardinal fish, their population is declining today The fact that sea urchin, which have been a symbiosis, are reduced because they are caught by the public for consumption. In 2010, in Matano Lake, we found an invasive species case, flowerhorn or lohan. Flowerhorn is an ornamental fish that has been gaining popularity in Indonesia since the 2000s. Fish from the cichlid family are the result of crossing from several genera, Cyclasoma, Amphilophilus, and Paraneterophilus. This fish is introduced by reason of hobby for anglers who often fish in the lake. Unfortunately, this fish is a predator that is very dangerous to the survival of native fish in Lake Matano Lake. We could imagine like putting a shark in a cage filled with snapper will be a slaughter and disaster. The second problem is a habitat damage. In 2009, we made an expedition to the La Susua River in North Kolaka, southeast Sulawesi. But in January 2010, a flash flood occurred and a decline in the type of fish found in the river based on the community report. Spots that have a lot of fish are decreasing, and today, unfortunately, mining is increasingly developing in the North Kolaka area, and we fear that it will further disrupt the preservation of native fish in the river. One obstacle we face in ornamental fish research, especially when collecting fish from its natural habitat, is the occurrence of death during transportation. This is immeasurable loss, the sacrifice of energy, toil, material, and time, with the number of fish produced from the collection process is really not worth it. As an example, in 2017, from 842 fishes we collected from Papua, only 40 safely arrived in Depok. This is very pure, very pure. After the fish have been collected from nature and ready to be treated in our research institute, there are still some obstacles we face. One of the most fundamental factors to be resolved is the determination of the sex of the test fish. But sometimes we encounter fish that have sexually monomorphic characteristics. What is sexual monomorphism? It is a fact which a species show no differences between male and female other than their genital. They have no secondary sex differences and no real differences in size, color, eating habits, etc. You know, the knowledge of fish gender will allow us to constitute mating couples or variable sex ratio in different experimental breeding group. There is a bias also in species that have a monomorphic character in which steroid hormones do not provide reasonable pattern, such as a high level of testosterone in male and estradiol in female. But in Papuan arowana or Sclerophagus jardini, testosterone was higher than estradiol concentration in all tested fish. Even in one of the dissected female, it was seen that the concentration of testosterone was slightly higher than estradiol. As you seen in the chart, this is the oocyte of the fish and this is the testosterone concentration. And in Arowana pino or Pino arowana, Sclerophagus macrocephalus, the variation of estradiol and testosterone fluctuation which observed could not be used as a reference for understanding the differences of male and female yet. One confusing case was that in one of the dissected male fish, the concentration of estradiol was much higher than testosterone. This Fumedi et al. Said that quantify of morphometric, body slenderize, head mulberry, fatness, and length of pectoral fins show nothing significant differences between male and female arowana. We've been done a lot of steroid measurement on this monomorphic fish, for example, tiger fish, Datnodius macrolepis, and fire eel, Mastasemblus erythrotaini, both using radioimmunoassay initially and recently using enzyme-linked immunosorbent assay, even if we still confuse whether it because endocrine disruptor compounds or the real concentration. We have tried to solve this monomorphism problem through the phytogenin assay approach. As we know that the PTG or phytogenin is an axial precursor protein, which is a specific characteristic of female protein, which means detect PTG also detecting that the fish is female. We've been stimulate PTG production in various species and also purified the PTG content. We have also tested cross-reaction between PTG and anti-PTG of several species, but still in laboratory scale. In 2014, Sansui et al. Proposed a patent related to a method of determining the sex of Indonesian red arowana by molecular approach. This is, of course, is an accurate method, but still in laboratory scale. In 2016, we developed the coagglutination test method, which is one of the fast and accurate method, but still constrained to be brought to the field, considering that the bacteria, because we use a Staphylococcus aureus, the bacteria require special care to remain active. This is control or without VTG. This is positive control with VTG. And this is unknown fish which has sign the same as a control, and this is a unknown fish which has sign the same with positive control. So we can conclude that C is male and D is female based on this coagglutination test. But once more, I say that it's still laboratory scale. In 2017, we proposed a patent for a PTG-based arowana sex detection method, but we realized that this kit is still for laboratory scale. This is just to show an illustration how to solve one of the obstacle or problem we face in ornamental fish research and still being developed till nowadays. Also, the problem is quality decrease because it's no longer in its natural habitat. Of course, one of the obstacles we face is also a decrease in the quality of the ornamental fish. The decrease in quality, both in terms of reproduction, also in color quality. For example, tigerfish until now has only produced eggs and sperm, but no fertilization has occurred. Various types of water and substrate have been used, but still the results are still zero. In color quality, test fish always experience color degradation compared when it's in nature. Of course, this obstacle is very closely related to the change of environment and nutrient intake. The obstacle face, the problem that also we face in research is parameter measurement. In monitoring is a diagnostic kit used on average still rely on imports. So this has an effect on the difficulty of the continuity of observation. These imported kits cost high price. For example, for the estradiol kits for it has price 5.6 million rupiah for 36 till 41 samples. This will be trouble if observations are made for very large samples. The specificity factor is also take some effect. For example, estradiol kits that are specific to fish, the price range from 18 to 25 million rupiah. Delivery factor, the range from 6 to 10 weeks, also cause difficulty analysis, carry out spontaneous when a case occurs. The length of delivery will also cause some time kits that arrive are not optimum due to the length of delivery and exposure to room temperature for a long time. So for a summary, we observe that the problem in research occurs in three phases. When in nature, it is related to community awareness, habitat destruction, and fish transportation. And at the research facility, we face constraint of sexual monomorphism, decreased quality and measurement parameter. What I didn't talk is about post-cultivation or post-research. related to transfer technology and marketing, because these require more time to discuss. To increase public awareness, we recommend at least the following idea. National movement ornamental fish display on every public space. So every government agency, airport, tourism site, there is an aquarium to display ornamental fish, so that the people are led to familiarize themselves with ornamental fish. And second is the national movement of one house, one aquarium. With this movement, we expect to form a community of people who have the responsibility for the preservation of ornamental fish. As we said earlier, that ornamental fish is not necessarily expensive, but depends on interest and depending on the type available in a particular area. Including for the aquarium, it doesn't have to be an expensive aquarium, even use food glass, jars, or anything. As influencer, we also need an ornamental fish ambassador who is truly active in carrying ornamental fish, not only based on an attractive face, but really because of his passion in ornamental fish. We also recommend a campaign for native and endemic ornamental fish lovers, so that people are more familiar with the native ornamental fish of the region than ornamental fish which is popular but introduced. Also, we realize that the largest income come from the export of introduced ornamental fish. There is an idea about the Banggai cardinal fish. One of the things that need to be considered in the future is research to anticipate policies as well as underlying policies. For example, for Banggai cardinal fish, we know that the Minister of Marine Affairs and Fisheries issued a decree number 49 / Capman Cape / 2018 regarding the implementation of BCF limited protection in February, March, and October, November. So in those months, it is necessary to ensure that the fish that are being exported are not from catching, but from aquaculture. Here, the role of research is needed, perhaps with a molecular approach to see the differences between fish caught in nature and fish from aquaculture. About arowana sex detection kit, I will show you the big number of arowana exporting each year since 2009 till 2018. Why this detection kit is needed? It needs to be realized that if the fish that are being exported constantly didn't pay attention to the sex of the fish, it is not impossible that foreign country will be able to cultivate this fish as well. And when they are able to cultivate, the dependence on ornamental fish from Indonesia will be reduced. And this will also have an impact on fish farmers in Indonesia. Therefore, the development of kit with detection methods that could be applied in the field, such as the lateral flow assay method, that is widely used in, for example, in pregnancy tests, is an effort to maintain Indonesian sovereignty, especially in the field of ornamental fish. In the future, if this kit has been established, it is necessary to make a regulation that only male okay. And for parameter measurement, the problem we facing in parameter measurement, overcoming the problem in parameter measurement, we initiate that domestic kits could be designed and produced domestically. This also allow us to design diagnostic kit as needed, and continuous monitoring could be done because the shipping process does not take long. For quality degrees, to improve, in addition to improving environmental quality and nutritional research, it is also necessary to continue to explore aphrodisiac compounds and stress regulating compounds. With this, we hope that ornamental fish research will be easier to achieve its goals. Here we see a possibility of relationship with head shock protein stimulants, which will be delivered by Dr. Charles and Mbak Marta. And the last, being together will certainly be better than being alone in goodness and truth. So the collaboration of all ornamental fish stakeholders is urgently needed. What could be done in collaboration? Inventory the native fishes in each location. And if there is potential ornamental fish, we may locally adaptate the fish or in situ adaptation. It is also better if there is a showroom in every potential site or location. For example, a lake or marine park, so the people know the potential of the site without dive or observe by themselves. We call all of you to concern about this. For example, in Matanolek, Islu, South Celebes, there is a mining company there, Vale. Vale could participate in build Matano Lake showroom. So there will be living specimen of each species in the lake, which rear in the showroom. To maintain it, we could empower fishery students in each region. In addition, the showroom could also be used as an intermediary tool when researchers need fish collection in the area. No need to survey for days long to collect the fish or at risk of death due to lack of adaptation time before fish transportation. And of course, if possible, to develop it into a museum. Besides that, we could also collaborate in disseminating technology, market promotion, and even conducting collaborative research. I would like to close my presentation with one verse from the Holy Quran that Inna Allah la yughayyiru ma biqaumin hatta yughayyiru ma bihanfusihim, that Allah will not change the condition of a people until they change what it is in themselves. So no one will make Indonesian ornamental fish move advance if not us ourselves. Thank you. There is a motto, yakin usaha sampai, which means when we effort, we should believe that we will arrive our goals. Thanks to a lot of parties that contribute in our work, include the participants that could be mentioned one by one. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for Pak Musa, Mr. Musa. It's informative and interesting talk. I wrote the important thing, like the background of the research, ornamental fish, because the unique of your ornamental fish, endemic, endangered fish. Some hobbies need the ornamental fish, habitat damage, and then fish genetic until the product are arowana insect detection kit as the product of research center for ornamental fish culture. Again, I give short information after the second presentation and the third presentation. It will be question and answer session. So please write on the question. Our team will be selected to read after the third presentation. Okay, the second presentation by Dr. Charles Saliba. His talk is about the role of heat shock protein and journey into HSP stimulant finding. Before the presentation begins, allow us to introduce the presenter. Charles Saliba is an entrepreneur with a strong technical background in the area biotechnology with more than 30 years experience. After graduating in applied biology and undertaking the thesis in the vitro modeling, He worked in the agribusiness and life science sector, setting up the number of research institutes and production facilities in various countries. I think include Indonesia countries. He also lectured at University of Malta and has supervised over 40 theses focusing on stress-related issues in animal welfare. Problem? It sure already published several international publications in journals. All right. Without further ado, Dr. Charles Saliba, the floor is yours.
Good afternoon. Can you see the screen and hear me?
Yes.
Okay,
Yes.
thank you.
We see.
Good afternoon to you. I would like to thank the organizing committee for giving me this opportunity to share some of my thoughts and my work with you, and also for the participants to give us your time to be with us. It is a pleasure for me to spend some time together with you, and hopefully we can have some exchange at the end of the session to share our ideas and learn from each other. I strongly believe that the way forward, I strongly believe that the way forward to increase the knowledge on a global scale is by sharing. If we share our ideas, if we share our thoughts, if we share our results, this will help every individual and society in general. So I really do welcome the opportunity to share ideas with people whenever this is possible. What I'd like to do this morning in the time that we have is to share some of our thoughts around the heat shock proteins and focusing on aquaculture. I will not delve too much into aquaculture. What I'd like to do is to discuss, hello? Can you hear me?
Yes.
Okay, is to discuss more heat shock proteins in general and just give some very small examples of the importance of heat shock proteins in species other than just fish, because I know there'll be other speakers who will spend time to discuss this in more detail. Hello?
We hear you.
Yeah. Can you see the screen okay? Because it's stopped moving. Okay. So the areas which I would like to go into and discuss is welfare, animal welfare in general, give some examples about heat shock proteins in aquaculture, discuss heat shock proteins and their importance, and some applications in human, in poultry and rabbits. And this will give us a bit of an overview of heat shock proteins in general. Traditionally, we would like to have good welfare for our organisms. And one way of having good welfare is to realize if our organisms are not in a good situation. So how can we tell that the organism is perhaps suffering from some kind of stress or is not in a good welfare? And traditionally, we used various techniques. So for example, if our fish were not eating properly, we would think that there is something that is actually stressing the fish, and this could lead to poor welfare. So it's a very important observation, but it's not necessarily always the case. Reduced growth very often follows reduced feeding. If the fish are not eating their total ration, they don't have the energy to to grow and function metabolically. So we see reduced growth. So this is another indication of stress leading to poor welfare. And behavior. We look at the behavior, how the fish are swimming, etc. And very often, if we see the panting, the fish are panting or accelerated respiration, we would associate this with stress. So we think that the fish are stressed, and this could possibly be an indication of poor welfare. So we used to use traditionally physical observations to determine whether our fish are stressed or not, but it's not always the case. Just because the fish may be exhibiting some of these or all of these parameters does not necessarily mean to say that the fish are stressed, or inversely, if the fish are not exhibiting any of these parameters, it does not necessarily mean that the fish are not stressed. So as time evolves, and we are doing this on a continual basis, we need to look at other techniques, at other methods of determining whether our fish are stressed, and could this lead to poor welfare. More recently, let's say the last 15 to 20 years, we started collecting samples, either tissue samples or blood samples, and more recently, water samples. And we measure cortisol. Cortisol has been used quite extensively as an indicator of stress. If there is a high cortisol level in any of the tissues we examine, particularly blood, we generally think that our organisms are stressed, the fish are stressed. But cortisol can increase for other reasons, such as, for example, if the fish are becoming sexually active, or if they're adapting to an environment, then they will elevate their level of cortisol. So just because we detect a high level of cortisol in our fish, it does not necessarily mean to say that the fish are stressed and they have poor welfare. So again, it's another indicator, not just behavioral, but there are possible situations where cortisol by itself is not necessarily a good indicator of stress and welfare. So as things are evolving, we are using a more holistic approach now, especially with the emergence of new tools. So we're looking at behavior, how are fish swimming, and how are they breathing, the physiology. Do our fish have lesions? Is the color correct, or are they losing color? Do we see abrasions? Are the fins damaged? So the actual physiology of the fish can give us some indication. Genomics is beginning to play a very important role, as well as proteomics. So as these new tools come into play and they become more accessible to people and more affordable to use, we can look at stress genes and which genes are being expressed at any time, upregulated, downregulated. So we have a better indication of what's happening on a genomic level. And also, are our fish susceptible to stress? So we can look at the genes, and some genes will let us know whether the fish are perhaps more susceptible to stress or less susceptible to stress. And we are beginning to see the emergence of two new areas where we are looking at either stress response pathways. So how are our fish responding, which pathways are being stimulated, as well as new biomarkers. So rather than just depending on a cortisol level, we're actually looking at a molecular level, what is happening inside the fish to determine whether our fish are stressed or not. And if so, can we do something to alleviate that stress? Because it's all well and good to have a tank, have an aquarium, have a pond, have a fish net, and you do all of these tests and you say, yes, I've done all of these tests. I've used the holistic approach based on the 5 parameters that I have just indicated, and they all suggest that my fish are stressed. The next question is, can I do something about this? Can I protect the fish? And can I bring about a good welfare state? And this is the approach that we took some 20 years ago to see if we can perhaps help by looking at the nature and seeing what the nature does, and then transposing some of that nature. So if we come a bit closer to home and start to focus, stress in aquaculture can be defined as any stimuli or stressor that threatens the homeostasis of the organism, resulting in either a neuroendocrine, a physiological, or behavioral alterations. So in very simplistic terms, any condition that can change a physiological, biological, or behavioral change in fish can be defined as stressed. And I'm not going to teach you this. You will all have very good experience, I'm sure, of the kind of situations or challenges that can induce stress on fish. And these vary from physical, such as temperature changes or radiation, particularly in ornamental fish living in aquaria. If they're too close to the lights, you can affect the retina in the fish, you can affect the color. Feed deficiencies, we are feeding fish artificial diets, and perhaps not all of these diets are the best to simulate the situation in the wild of our farmed fish or our aquaria fish. Oxygen levels, Mas Musa described the issues associated with transportation from the wild, a very big issue in transporting fish in any situation, but particularly wild caught fish, is the level of oxygen. Fish are very susceptible to low dissolved oxygen. Perhaps less so in ornamentals, but the stocking density, because in ornamentals we generally use low stocking densities, but in other species we use high stocking density. And the list goes on: transport, algal blooms, various biological challenges, bacteria, virus, parasites, genetic abnormalities. All of these parameters can induce a stress on our fish and lead to poor welfare. So in our work, in my institute, in my organizations, we look at nature. Today we have become very accustomed to using pharmaceutical products to help us, but the approach that we use is to look to the past in order to try and see what can we do for the future. And we develop what we are referring to as novel alternatives. These are products or activities or way of farming that will help our organisms, our fish, to cope with the stressful situations they may be in based on nature. How does nature do things? And can we try and reproduce the way things are done in nature? And one very important parameter that we need to remember is that phytochemistry is the forefather of pharmacology. Very often we don't appreciate this, and we think that pharmacology is the be-all and end-all. But in reality, this is not the case. So if we look at the Chinese traditional medicine, the register has been around for around 1100 years. So perhaps the use of novel compounds from nature is not so novel after all. And in our part of the world in Europe, Hippocrates was making cures for various ailments, including stress, around 450 years before Christ. So perhaps the idea of using novel compounds from nature is not so novel overall, after all. So what is our objective? What is our target? So what we try to do is to look at the nature and come up with solutions that target various points in the organism to bring about welfare. And one area of focus is the immunity. Can we help the organism to mount a good immune response, very often mediated by T cells, in order to allow it to identify challenges, biological, viral challenges, very early on, mount a strong immune response that will last for a long period of time to help it through the biological challenge period. We can also look at hormonal changes. How does the organism respond to stress or to, shall we say, challenge, and challenge can come in many shapes or form. What are they doing on a hormonal level? And similarly, on a physiological level. Can we do something to help the organism to protect the integrity of its membranes, the integrity of its cells, the integrity of its DNA, in order that the physiology is not affected or damaged? Thus, the organism can cope with the stress and have good welfare. So this is our mantra. This is what we are aiming to do. And we try to do this using natural tools. And if we look at nature, a tool that is used very extensively by nature in all species are heat shock proteins. Heat shock proteins are very, very ancient biological tools. They are present in all eukaryotic organisms. And their main role in the nature is to regulate protein synthesis and protect membranes. So they are at the core of protein synthesis. So they regulate the folding of newly synthesized proteins. They protect the integrity of nuclear matrix and matrix material. They repair. Sometimes proteins get damaged and they don't function in the manner that they do. So heat shock proteins come in and they repair the altered proteins. Another name for heat shock proteins is chaperone proteins or chaperonins. And this name came from the idea that you have these proteins, chaperonins or chaperones, that will actually chaperone, they will guide the protein synthesis in order to make good protein that is functional. They're involved in transportation of proteins within the cell, between organelles, and between cell to cell, through cell junctions. So they're very important in the transportation of proteins. And they're also important in the elimination of damaged protein. If a protein is damaged and non-functional, it is a burden on the cell. So the heat shock proteins will eliminate the damaged protein from the cell. So how can we go about do something about heat shock proteins? Can we do something if there is a stress, if there is a demand for the cell to synthesize heat shock proteins to protect its own proteins? And we do this by looking at nature. I'm sure that most of you are very familiar with this beast. It's a cactus. In English, the name is prickly pear cactus, and the Latin name is Aquaculture ficus indica. And we looked at this. What attracted us to this is the actual plant itself. This plant defies nature. In our part of the world, this plant produces the fruit in August. August in the Mediterranean is the hottest month of the year. And where we are in Malta, in the center of the Mediterranean, and where Malta is in the south of Spain, temperatures can be 45, 50 degrees in the shade. In August, this is very severe because it's also very dry. Yet this plant decides to produce its fruit in August. And another thing that makes this plant a little bit different to perhaps other plants, such as grapes, for example, which also produce their fruit in August, is the position of the fruit. This plant has decided that it will expose its fruit at the uppermost part of the plant, exposed to the sun. This obviously, perhaps or not so obviously, made us think that this plant is perhaps not compliant with nature. Why should you expose the most valuable part of the plant, which is the seed, carrying the DNA for the next generation, to such harsh conditions? Why should you do this in August when it's very hot and exposed to the sun? The temperature inside the fruit in this part of the year is about 60 degrees. So a question that comes very quickly to mind is, how do you keep protein stable at 60 degrees? And the answer came relatively quickly, is because this plant can produce heat shock proteins, and it does this very efficiently. It can do this very quickly, within minutes. So this is one way that the plant can protect its own proteins. So with this in mind, we try to see if we can make an extract from this plant in order that we can transpose this activity that we see in the plant, that the plant is capable of synthesizing heat shock proteins, and use this in other species. So this is what we set to do. This was our target. And we've done close to 20 years research, and we've had numerous publications. When we first started doing this work, there was no ELISA available to measure heat shock proteins. So we had to develop our own ELISA. We made our own primers, we made our own antibodies, and we developed our own method of analysis, not just quantitative, but also qualitative heat shock proteins in cells and organisms from various types of organisms. At that time, there were very, very few publications around heat shock proteins. Today, it's a huge area, and if you go to the literature, there's over 500,000 publications around heat shock proteins. We focused on a number of species, but we have, thank goodness, a very strong team of researchers around us, and some of these papers, these are just a small example of some of the publications we've had, let's say, in the last 5 years around heat shock proteins in aquaculture. So heat shock proteins are becoming very important in aquaculture, and there are many researchers today all over the globe looking at the role of heat shock proteins in aquaculture. How can we modulate heat shock proteins in aquaculture for a number of benefits in aquaculture? And what I hope to do today is, rather than focus on aquaculture, is give you some examples of other species. Mas Musa discussed also the importance of fertilization, or shall we say reproduction. It is very, very important that we can reproduce, and I will talk slightly, briefly, about the work we've been doing at improving fertilization, or shall we say fecundity in various species. And my colleague will also discuss this in more detail, how this applies to aquaculture. And we'll touch about this later on. A bit closer to home, koi, and I would like to salute my old mentor, a very eminent person, perhaps one of the first people that discovered aquaculture or set about aquaculture, and one of the first researchers in aquaculture and heat shock protein, probably the first ever, Professor Ron Roberts from Stirling University. So I would like to salute him in this presentation. And we've done some work with him also in koi, and we'll be discussing some of this work later on. So if we look at heat shock proteins in relatively simplistic term, what's happening? If an organism, if a fish is exposed to a number of physical, physiological, or biological challenges, that organism detects there is a stress. And it does this by observing the oxidative level inside its organism, inside the organism. The oxidative level will change. The fish detects that there's a change. The oxygen demand by the organism changes. This will induce an oxidative stress inside the fish. And the fish needs to do this because very often stress brings about the elevation of raw species, reactive oxidative species within the organism. And these ions can do a lot of damage to very delicate proteins, cell membranes, nuclear membranes, etc. So the ROS activity and the oxidative level of the fish detects the stress and brings about an HSP response. So when there is a change in the redox state of the fish, the mechanism for synthesizing heat shock proteins is activated, and the organism will be able to cope with the change in the environment, with the stress, by elevating its level of heat shock proteins. What we discovered very early on as well in our research is that the heat shock proteins are universal. So if we look at heat shock protein 70 in a fish, an ornamental fish, or a tuna, or a salmon, or a dog, or a shrimp, or a poultry, or a cat, or a human being, it's the same heat shock protein. So heat shock proteins are universal. So if all eukaryotic organisms have heat shock proteins and they all have the same heat shock protein. Can we use the extract that we produce from this wonderful plant that perhaps is not so wonderful if you fall into it because it's full of spines, in order to bring about a heat shock response in all of these type of organisms and help the organism to cope with some form of physical or physiological stress? So this is what we set about to do and to try and demonstrate if we can show some benefit by doing this. So what we've been able to do is that by demonstrating that through the use of this extract from this plant, we can bring about a very fast heat shock 70 response, not just heat shock 70. We've looked at the family of the 90s and the 20s, but we focus mainly on the family of the 70. And we've demonstrated that it is possible to prevent oxidative stress. It is possible to prevent the damage caused by thermal stress, osmotic stress, and desiccation in aquatic organisms. So we have a tool that's coming from nature. It's a total extract from an edible plant that has been demonstrated to have a number of benefits during stressful situations in aquatic organisms. And my colleague Marta will describe this in more detail. What I'd like to do is demonstrate how universal the heat shock protein response is. So we looked at humans, and can we see heat shock proteins in humans? And can we measure these heat shock proteins in humans? And if humans are exposed to a stress, can we give them some benefit through the role of heat shock proteins? So we set up an experiment. We took some subjects. We split them into two groups. Half of the group received a placebo, and the other half received the extract of the cactus. And we exposed them to a stress. So we put them in a sauna every day at 85 degrees for 15 minutes on 3 consecutive days. And we measured their heat shock proteins. And then 3 weeks later, we split the group again, and the people that received the placebo in the first set of experiment received the extract, and vice versa. And we repeated the experiment. And at every stage, we measured the heat shock protein. So each person acted as their own control. And what we observe is that at the start, the level of heat shock protein, there is a level of heat shock protein in all the subjects. Under the control situation, when they receive the placebo, if there is stress when they go into the sauna, it takes about 2 hours to elevate the level of heat shock protein, and the level diminishes within 24 hours. But if there is subsequent stress, they go into the sauna every day, the ability to increase the level of heat shock protein diminishes. And on the third day, it's even less. But when the subjects ingest the extract of the cactus, what we observe is that there is an increase in heat shock proteins within 30 minutes, and the level of heat shock protein remains high for 3 days, even though these subjects are being exposed to stress on a daily basis. So we were able to demonstrate that, yes, humans have heat shock proteins. When they undergo a thermal stress, they elevate their level of heat shock protein. And by ingestion of the extract of the cactus, it's possible to get a much faster, much more rapid heat shock protein response, and a much longer, much prolonged high level of heat shock protein response, even though the subjects are exposed to a number of stresses, a thermal stress in this particular case, on a daily basis. Our team is very closely associated with divers. All our people are divers. This is important for us for a number of reasons. One, we work very closely with aquaculture, so we need to go and look at our fish. We actually dive with the fish, we swim with the fish to see actually what's happening inside the tank very often. We also work on a number of marine algae where we cultivate algae and we collect algae. So we are all divers. It is important for us that we dive on a regular basis, not just to keep our sanity, but also to do our work. So what we try to do is to see what happens during a dive. A dive is a very stressful episode. The diver is exposed to a number of physical and physiological changes. Very often they get hot or cold depending on where you're diving. You're doing exercise, so you're burning up energy. You are under pressure, and this has a number of factors. You are forming microbubbles of nitrogen, and this can be very dangerous if not properly controlled. So the diver is exposed to a number of physical and physiological challenges during a dive. So we try to simulate a dive inside the diving chamber where the divers are actually sitting down, and we can collect blood from these divers at specific points in the dive and measure the level of heat shock proteins. So one observation is that at the start of the dives, generally the divers are quite okay, but at the end of the dive, the divers are tired, they're fatigued. Now keep in mind, these divers, all that they were doing is sitting in a diving chamber undergoing a simulated dive. They have not done any physical exercise at all. They have not got wet, they have not put on a wetsuit, they just sat in a diving chamber to simulate a dive. So if we look at what's happening during the dive itself, so this is the dive profile. So they are at the surface, they dive to around 35 meters. They spend around 30 minutes at the bottom of 30 meters. They surface. After 30 minutes of diving, they do their decompression stop at 5 meters, and then they come to the surface again. A standard, what we call box dive. You go to the bottom, you stay on the bottom at one depth, you come up to the surface, decompress, and come out of the water. So a very standard, most people wouldn't consider this as a particularly hard dive, but just a standard dive. And again, as we did with this one experiment, we had a group of divers, half received the placebo and half received the extract. And then 3 weeks later, we do the same dive, but we cross over and we measure the heat shock protein in the divers. And what we observe is that when the divers take the placebo, the control, there is an increase in the heat shock level, but it takes 2 hours. Whereas when the divers ingest the extract, there is an increase in the level of heat shock protein within 20 minutes. How is this important to the diver? If the diver is to have an accident, generally the accident will happen in this part of the dive. The diver would have been diving, accumulating microbubbles in his or her blood, and as these bubbles start to expand, as the diver ascends, the pressure is less, the bubbles get bigger, and you can get an embolism. A bubble will be big enough to block a vessel, and this can be very fatal. The diver can become paralyzed or even die. This is what we call diving sickness or the bends. So what we've been able to demonstrate in this experiment is that should an accident happen in this part of the dive, by ingesting the extract, the diver is fully protected before they even start their ascent. So by modulating the rate of synthesis of heat shock protein, we can protect the divers against the possible consequences of a diving incident. So this is an example where we can use the modulation or the regulation of heat shock protein to bring about some protection to an organism. Poultry. Poultry is another important species. It's grown all over the world. It's possibly, along shrimp, the #1 protein that is eaten by people on a global scale. So poultry is very important. So we looked at poultry again, as we did the human, and to see if we can demonstrate heat shock proteins and some benefit. This work started about 7-8 years ago now, and at that time, no one had published any work at all regarding heat shock proteins in poultry. We were the first people to publish a paper demonstrating that poultry do have heat shock proteins, and also it's possible to modulate the rate of synthesis of heat shock proteins. And when you do this, there are some very clear benefits. If there is a stressful situation, an upset to the homeostasis of the organism, by applying the extract of the cactus, we can modulate the heat shock protein to bring about some benefit to the organism, to the poultry. So we set up a very, very basic experiment in chicks. So if we look at a chick, the first day of life is an incredibly stressful period. The chick has been very comfortable sitting in its egg in an incubator. It's warm, it's biologically stable, it's sterile. Suddenly it comes out of the egg and it's faced with a very hot incubator, no water, no food, and exposed to biological challenge. It is then taken out of the incubator, handled, physically handled by people. It's vaccinated, put in a box and put on a truck, and can get very cold or very hot, depending on where it is. It's driven to a farm, and that's put into a shed, and then it has to find food and water and compete for that food and water with its brothers and sisters, as well as cope with the biological challenges that are found on the farm, because the farm is not sterile. So the first day of life for a chick is a very stressful period. So we set up an experiment where we took some newly hatched chicks as soon as they hatch, and we collected some blood from them. Then we applied the cactus extract. So we sprayed the cactus extract on them, and they ingest it through the nasal cavity or the ocular cavity through the eye. And after 1 hour of spraying, we collect some more blood from these chicks, and then we take them to the farm. And the following day, we collect some more blood from these chicks to measure the heat shock protein. And we have obviously treated chicks and control chicks that were not sprayed with the extract, so we can measure the response in heat shock protein. And what we observe is that there is a base level of heat shock protein, so we were very happy that we could find heat shock proteins in chicks. And what we also observed was, as we've seen in aquatic organisms and human beings, that within 1 hour, these chicks could elevate significantly the level of heat shock proteins. And 24 hours later, these chicks still had a very high level of heat shock protein in their system. But the control chicks, the chicks that were not treated with the extract of the cactus, they could not elevate statistically significantly the level of heat shock protein within 1 hour or within 24 hours. So even though these chicks are being treated in the same way, they're being exposed to the same stressful conditions, welfare harming conditions, shall we say, these chicks, the control chicks, were not able to elevate their heat shock proteins, and consequently they were not able to respond. And we've done data, I won't show, it's not the purpose of today, but we have data today on over 100 million birds, where we have demonstrated the benefit of such applications in terms of their growth. So we can improve their growth. We can get perhaps on a particular type of bird on a Ross 308, an extra 50 grams of live weight by 36 days. We have a better FCR by about 5 points. We reduce the mortality by about 0.5%, and the welfare of the bird. So there are what they call foot lesions in the birds and heartburn, where these birds are being challenged by bacteria which cause lesions. We can significantly reduce the incidences of these conditions. One thing I'd like to stress is that when you're doing this kind of work, in order to have good statistics, you need many replicates. So we always work with large numbers. So as Mas Musa was describing, the access to good material to do analysis is key to all of this work. As you can see, we analyzed 600 blood samples for this experiment. So it's not a few samples. Can we demonstrate a benefit of this early increase in heat shock proteins? We work very closely with veterinarians on a global scale. And there was an incident where a veterinarian was called to examine some birds. These birds had been vaccinated with Pastorella vaccine. This is fairly normal in order to protect them. The birds are grown on a rearing farm before they go onto the on-growing farm. And the farmer was a bit concerned because his birds were not eating properly. Again, biological and behavioral changes that we see, and they were not growing as they should. They were not gaining the body weight that they should be gaining and eating as they should. So the veterinarian was called out to see these birds. This is fairly typical after this type of vaccination. But when the vet went to see the birds, the reaction to the virus was perhaps a little bit more severe than it would normally have been. And the symptoms were very clear, swollen heads and swollen necks. So the vaccine had induced a huge inflammation, a huge immune response, and the bird had a lot of tissue damage. The area around the granuloma where the injection took place for the vaccine was very severely damaged. There was a lot of necrotic tissue in the area around the granuloma where the bird had been vaccinated, and there was vaccine deposit all along the breast muscle. So the muscle itself was being damaged. So a lot of physiological damage being incurred on these birds, apart from the fact that these birds were eating properly or growing properly. So again, very clear stress markers that the birds are really undergoing some form of stress, and there is going to be a welfare issue unless there is some form of intervention. So the vet gave these birds some cactus extract through the drinking water the next day, and he repeated this 4 days later. And within a day after the first treatment, the birds started feeding again and gaining weight. After the second dose, 4 days later, they carried on to grow in a very good manner. And 7 days after the first signs of the vaccine damage, we carried out postmortems on these birds, and we saw that there were no signs again of the vaccine damage, and the birds could be moved on to the on-growing farm. They could have been sold. Had this intervention not taken place, those birds would not have been able to be sold on to the farmer, and they would have probably had to be sacrificed because they were not growing properly and reaching their target weight. So we can demonstrate in real terms on an industrial scale how the modulation or the regulation of heat shock protein can be of benefit to organisms. Another model, really for demonstration purposes, but this also has very significant industrial applications, is breeding. A term that we have in Europe is they breed like rabbits, which goes to imply that rabbits can have many, many babies. In industrial production of rabbits, this is not always the case. And fertility is a very important issue because you are growing your adult rabbits, and the main purpose of growing them and feeding them and looking after them is to produce more rabbits so you have more food. But if the mating does not lead to fertilization, which does not lead to the production of more rabbits, then the whole farming operation will crash. And believe me, a number of operations do suffer where the rabbits do not breed as well as they should, so the farming does not become economic anymore. So we looked at a large number of rabbits to see if we can have any benefit, and we applied the extract to upregulate the heat shock protein in these rabbits in the drinking water of the male and the female. And we also rabbits, there's two methods of fertilization in rabbits: the natural way between the rabbit and the doe, and the artificial insemination, where we collect sperm from a prime rabbit. We analyze the sperm, we dilute the sperm so we can fertilize many females from the sperm of one male using artificial insemination. So we did a number of experiments with over 3,000 does, either giving the cactus extract to the drinking water and using natural fertilization, or adding the cactus extract to the sperm directly and then fertilizing the doe with the sperm that had been treated with the cactus extract. Again, sperm is a very important tool in fertilization, and the sperm, in order to be able to fertilize an egg, has to go through a lot of physical and physiological changes, especially in artificial insemination. It is not in its normal environment, it's taken out of the animal. Very often it's diluted, and the sperm has to be reintroduced into the female, swim in the vaginal tract, find the ovary, and fertilize the ovary. This is a huge energy demanding process on the sperm, as well as compete with other sperm to get to the ovary first. So there is a big energy demand, there is a big change in the physiology around in the environment, so it's a particularly stressful period for the sperm. And unless the sperm is capable of fertilizing the egg, fertilization does not occur, and we do not get the baby rabbits being developed. So as I indicated, we looked at both natural and artificial insemination in does and 3,000 does. Some of this work was carried out in Malta, some of it was carried out in France, and so we could measure the efficiency. And what we see is very significant changes, or shall we say increase, in the reproductive rate when the rabbits are either treated or ingest the cactus extract. 20% in fertilization could make a difference between a farm being economical or not. So very, very clearly, we could demonstrate the benefit of modulating or upregulating heat shock protein in fertilization. So in summary, we can say that heat shock proteins are very ancient biological tools, and these are common to all eukaryotic organisms. The application of cactus extract enables the organism to elevate, to increase the level of heat shock protein within 20 minutes following a physical or physiological stress, and maintain a high level of heat shock proteins for 3 days. This will allow it to cope with many challenges. So the early and elevated prolongation of heat shock proteins has been demonstrated to protect in organisms from various species during periods of physical, physiological, and biological stress. And I really would like to stress this point and perhaps share my thoughts and ideas with you that we really need to look at the nature. Mas Musa was describing the importance of preserving the ornamental fish environment and promoting the environmental fish environment. This is a natural environment. And I would like to invite all of you to share with me this concept that we look at the nature and see how nature copes with the challenges it encounters, and by mimicking or trying to understand, it's not just simply copying, understand the mechanisms, the biological, the molecular, the physiological mechanisms that these plants do to cope with stress. Perhaps you can learn something from them that will help us, benefit us, our children, and our future generations. And I would like to thank you all for giving me this opportunity and your time. Thank you.
Thank you. Can you hear me?
Yeah, thank you.
Okay, thank you, Dr. Charles. It's informative, interesting talk, many information about aquaculture stress factor, many novel alternative to solve several stress factors. I think we continue to third presentation. Marta Arredondo Lucky, Master of Science. Marta will talk about application of HSP stimulant in aquaculture industry. Before presentation, I would like to introduce Marta. Marta Arredondo was graduated in Food Science and Technology by University of Cordoba in Spain. followed by Master of Science in Sustainable Aquaculture by the Institute of Aquaculture of University of Stirling. Her main research interests are the functional diets in aquaculture, focused on stress, parasite, and mycotoxin control. She is technical commercial responsible for the aquaculture department in Tecnofit Farm Phase in Spain. Okay, we continue. Marta, the floor is yours.
Lost my... Hello, can you hear me? Can you hear me?
We hear you clear.
Okay.
Okay. We hear you.
Thank you for attending the webinar. Also, I would like to thank the Research Institute of Ornamental Fish in Indonesia and Mas Musa for inviting me and myself and Charles. My name is Marta, and I'm the technical commercial responsible for the aquaculture department in Tecnofit Farm Faes. And I will be talking about the application of heat shock protein stimulants in aquaculture industry and also in ornamental fish. I will go through some of the trials we've carried out in several species, such as ornamental fish, sea bream, shrimp, lumpfish, or salmon. These are just some of the trials we've carried out. There's many more. I would like to start by talking about how heat shock proteins model work mainly in aquatic species. I would like to talk about two main examples, such as cross-tolerance and immunity. How this works is basically heat shock proteins can prevent the thermal stress and also protect against osmotic stress. A very good example of this is cross-tolerance, because it's a process where an organism, in a case of, or in this case is salmon, which you can see in the picture, is exposed to some sort of physical or physiological stress, which is the increase in temperature, which is now due to climate change quite normal in all aquaculture industry. This will induce an increase in heat shock protein level. And this increase in heat shock protein level will help induce the fish to have an osmotic resistance to this physical and physiological stress. In the same way, a biological challenge, for example, can also induce the heat shock protein synthesis. Normally, in most hatcheries, in all species, the conditions are controlled, sterile, and they are completely controlled, and we can make sure that the fish or the shrimp or the whatever organism, even the ornamental, the conditions are controlled. But when you put them out into ponds, or you transport the species, or you put them in cages, in the case of salmon, you cannot control all the conditions. It's quite challenging to have a sterile or a controlled environment. Basically, when the organism is exposed to this bacterial challenge or this pathogenic challenge, biological challenge, in this case, shrimp, which is a normal case scenario in shrimp farming, they need to be able to cope with the challenge, with the stressful challenge. And how they do that is they increase the heat shock protein level. And this can help them enhance the immunity they need to cope with that stress. How does this work, and why do we assume this is happening? Basically, heat shock protein play an integral part of the major histocompatibility complex, presenting the antigen to the adaptive immune system. And this is mediated through stimulating T cells. This is why there's an enhanced immunity that allows the destruction of the pathogen or help the compromised immunity of the aquatic species. So from these two models, we can say that heat shock proteins modulate both innate and adaptive immunity. And heat shock proteins are very important and have a very important role maintaining homeostasis, a good welfare state, and allowing the organism to cope with stress. For this reason, as Charles was talking about, heat shock protein stimulant was investigated and now applied to aquaculture industry and ornamental industry. And this, what it does, it accelerates the synthesis of heat shock protein synthesis. It enables cells to respond to the repeated stressors without exhaustion. And it's a natural cellular response to all eukaryotic cells. This is a patented active ingredient from Prickly-Pear Opuntia ficus indica, obviously non-toxic because it's a natural extract, and has various documented pharmacological properties. For this, for the aquatic species, the name for this product is called Ceretec, and I will be talking about the different trials we've conducted and I've chosen to present in this webinar today. Ceretec can be included both in feed and immersion. I assume for ornamental, the best way to include this product would be obviously by immersion because the tanks are not. Yes? Yes, Musa? Musa? Can you hear me properly?
Yeah, you can continue.
Yeah. The dose for immersion is 4 milliliters of Ceretec for every meter cubed of water, and the bottles are presented of 320 milliliters. For in-feed, it would be adjusted to feeding rate and biomass of the production. How does it act? Basically, you would apply the Ceretec only 30 minutes, from 30 minutes to 2 hours prior to the stress exposure. And what it does, it counteracts the physical and physiological stress, aids cell repairing, accelerates natural response following stress, promotes the return to standard growth profile and standard feeding profile, and the application of 1 dose of Ceretec will protect over 72 hours these aquatic organisms. So I will now present some trials we did in ornamentals, in angelfish specifically. As you know, for ornamental, this is a quite common scenario. I'm quite fan of having aquariums, and I've had aquarium for some years now. And this is a normal scenario when you have an aquarium or when you have fish in general. You just got some fish, and by the time you get home or you get to the tank, you've lost some of them. And this is due to stress. This is either by the low quality of the water during the transport or during the containment of the fish, because of the handling. It can be caused by different stressors. For the first trial I would like to talk about is low quality water. I will talk about nitrate, ammonia, for example, dissolved oxygen. Tropical fish are normally bred in land-based hatcheries using RAS, and these conditions where they are bred are quite are quite controlled. They have everything they need, the sterile conditions. But these fish are normally shipped to other countries, maybe to the other part of the world, Europe, United States. So during this process, fish are exposed to several challenges, including crowding, abrasion, stress, handling, changes in water temperature, changes in water quality. And one of the most important changes and stressors is water quality for fish, because that is their mean of living. So when the water quality deteriorates, it starts deteriorating. This happens quite quickly. And sometimes we can't control. During a transport, you can't control that much the water quality. In the case of Ornamental, most of them are shipped in big plastic bags. So you can't open and close those plastic bags every single hour or change water so often. So this in vivo trial was carried out with angelfish, and these angelfish were exposed to a high concentration of free nitrate during 72 hours. As you can see, in the non-stressed group with no Ceretec, the survival is 100%. The fish are not stressed, there's no problem. When the stress starts, we can see that the fish that were not preconditioned with Ceretec, there's a high mortality with an only survival under 40%, whilst the ones that were preconditioned with Ceretec have over an 80% survival. In the same way, following this trial, we analyzed also the liver tissue in those fish, seeing that those treated fish have a better condition in liver tissue than those of them that were not treated, because these fish that were preconditioned with Ceretec are allowed to have a better cellular recovery. In the same way, following the low quality water scenario, we also did another trial with ammonia, and these angelfish were exposed to 1.1 ppm of free ammonia during 72 hours. In the same way, we can see there's a high mortality in the fish that were not exposed to Ceretec prior the stressful period, which in this case is the ammonia, whilst the ones that were preconditioned with Ceretec have no change and seem not to be affected by this stressful challenge. But this is not because of magic. Basically, what this ammonia exposure has caused is a complete destruction of the lamella in the gill tissue. As you can see in the treated one, you can see the lamella are in perfect conditions, and fish can continue with their normal breathing, and there's no effect in oxygen available in the water, basically. Following the same ornamental trials, we also did the ethanolic stress exposure, and angelfish were now exposed to 5% ethanol during 30 minutes, only 30 minutes, and their survival when the time goes on decreases quite rapidly, reaching under 10% survival when it's over 24 hours, while those fish that were preconditioned with Ceretec have nearly 100% survival. This is because of heat shock protein synthesis. Again, we can see that those that were non-treated alive have some heat shock protein synthesis, and this is shown by fluorescence, in the muscle tissue, while those non-treated and dead have very, very low heat shock protein synthesis. Those that were treated with Ceretec have a high heat shock protein synthesis, helping this fish cope with the stressful period. In this case, it's ethanolic stress. Following these trials, I would like to show some of the trials we've done in sea bream. For sea bream, in the same way as I was commenting before, ethanolic stress is a problem because of anesthesia. Anesthesia is quite common when you are handling fish, and many anesthetic are not soluble in water. They are dissolved in ethanol prior to addition to a tank holding the fish, and also some anesthetics are based in alcohol. Alcohol is obviously a toxic substance for all fish, and that can do quite a harm to the fish. And also, if it's in high concentration or if the period is too long, it can cause high mortality. This in vivo trial, as well as the other one I showed in ornamental fish, show that this has a very harmful effect in both bream and ornamental. After only 15 minutes of exposure to 5% ethanol with an hour of pre-stress, we can see that the survival after 30 minutes is under 40%, while those fish that were preconditioned with Ceretec have no affection to their survival. This continues to decrease with nearly under 10% survival after 24 hours to those fish that were not preconditioned with Ceretec. A big stressor also in bream is the quantity of dissolved oxygen in water. So, for example, in bream, when there's a hatchery or in a transport, the dissolved oxygen is very controlled, and you can make sure that these fish have enough oxygen to be in good quality health and their welfare is okay. But when these fish are transported and put into sea cages, now the water temperature of the water due to climate change can provoke a reduction in dissolved oxygen, and this can be a big stressor for bream in sea cages. And you can't really control the dissolved oxygen in the middle of the water in I don't know how many kilometers from the coast. You can't really give more dissolved oxygen or increase the dissolved oxygen in those fish. So what it causes, it causes a big stressful challenge for these breams. And the trial shown here, which was done obviously in tanks, the breams were exposed to 0 ppm dissolved oxygen for 15 minutes, an hour pre-stress, and this caused a high mortality, which resolved after only 30 minutes after stress, under 20% survival, whilst those fish that were preconditioned with Ceretec have 80% survival, even 24 hours after the stress. For bream, we also did a big transport of blue stocks with a transport of 4 hours and 45 minutes with a 35 kilo per meter cubed stocking density. And during the transport, obviously, which is a normal case scenario, there's an insulation during transport, an increase in temperature. Then fish were put into recovery tanks. What we saw that there was a 9.3% mortality without Ceretec, as well as a deterioration of the skin, a high coloration of the skin, and 85% more lesion without Ceretec. I would also like to comment some trial reports we've done in shrimp in Ecuador. a big issue in when shrimp are produced is both deformities, intense intestinal contents, and molting process. We did some trials in hatchery, evaluating these three parameters in SOEA stage, showing that the shrimp that were preconditioned with Ceretec have fewer deformities, an improvement in intestinal contents, and obviously a decrease in non-molted shrimp. This is very important because shrimp that during the hatchery have these problems will probably deteriorate their performance during grow out. In the first stages, in the hatchery stages, the shrimp have to be in very good conditions, and the fact that they don't have deformities will improve the high quality standards in harvest. The intestinal content will help us cope with other stressful periods during the pond culture, and the molting process will improve the quality of the shell and will give a high value to those shrimp after grow out. In the same way, we did in the hatchery also a microbiological analysis. This microbiological analysis showed that those shrimp that were preconditioned with Ceretec have decreased their quantity of two of the main Vibrio species that are normally found in shrimp farming, as well as the Pseudomonas species were nearly halved in this hatchery shrimp analyzed. Also, we analyzed the growth parameters in post-larvae 24 and 31 days, seeing an increase in growth during the hatchery. Mainly what all aquaculture experts agree on is that during the hatchery in all aquatic species, if we are able to improve growth and if we are able to improve performance during these stages, this can help nearly double the improvement in growth and performance during grow out. And this is quite clear that the precondition with Ceretec can help the post-larvae improve their growth parameters. We also did a stressful challenge, such as a salinity test, showing that Ceretec can help a stressor such as salinity in shrimp. This is a quite important issue because of pond culture. Basically, in pond culture, you can't really control the salinity of a closed containment system, such as a pond. It's not like a rust or a flow through when you can control all the water body, because of the large water bodies we are dealing with and the changes in salinity that can be caused by big, heavy rainfalls or dry seasons. And this is also an important issue in hatchery and in grow out. We also measured the performance in grow out shrimp in Panama grow out conditions, showing that Ceretec continues to increase the weight while exponentially, while the shrimps that were not preconditioned with Ceretec from week 5 start to lower down the exponential increase. In the same way, we did a transport trial with Panama again, following elevated temperatures with nearly a 10 degrees increase from 25 degrees to over 35 degrees temperatures. And this shows that survival can be highly affected with non-preconditioning of Ceretec. In this case, those that were not preconditioned with Ceretec have under 40% survival, whilst a 50% survival is acquired when treating the shrimp and preconditioning the shrimp with Ceretec. This increase of survival during transport can make the difference when looking at viability of shrimp farms. In the same way, I would like to comment some lampfish trials. I don't know if it's common in Indonesia, but lampfish in Norway and Scotland are commonly used as cleaner fish, and it's a good sustainable strategy to cope with sea lice with parasite control. They basically eat off the parasites from the salmon. And this trial with lampfish was carried out in Norway in 2019. Basically, lampfish are fish that grow very quickly, so they're not that strong when they are put into cages. And they are very susceptible to bacterial challenges, to pathogenic challenges in general, and any water quality changes can cause high mortalities in lampfish. So we conducted a trial in Norway in RAS system. They are cultured in RAS system until they reach the size to be able to be put into the cages, because if they're too small, they will go through the cages, through the nets, and they will be lost basically in the wild. So to avoid that, you have to culture them in RAS system, and then when they are big enough, they will be put into the salmon cages. So in that RAS system, which has a high interchange of water, there was a natural bacterial challenge of Vibrio ordalie, which is quite lethal for lampfish. They had tried already with antibiotics, and we did a trial with 13,000 lampfish for control and 13,000 lampfish for the Ceretec treated tanks. Basically, what the control had the antibiotic they were using, which was not working as well as it normally did because of antibiotic resistance, we assume, and the other tank was preconditioned with only Ceretec. What this trial shows basically it gives a hint that Ceretec can help antibiotic resistance, such as Charles was commenting in poultry. In fish, we can also have an opportunity to help antibiotic resistance with, for example, Vibrio. And the control one was treated with fluorinphenicol in a daily basis, while Ceretec was applied twice a week. During these 4 weeks of treatments, mortality was recorded, and the control had over 5% mortality, whilst the preconditions with Ceretec had more than 1.5% less. 1% mortality. In the same way, I would like to give a hint on trials in salmon, which is quite common, and we have quite a lot of trials, but I would like to give an example of the trials that Charles was presented on rabbits regarding reproduction. This was also evaluated for salmon, and Ceretec was used in a combination as a sperm extender. They normally keep the control extender was kept with only PBS, and the Ceretec was the milt was kept with Ceretec. The score shows that the motility over the score of 1, you can assume that the viability of that milt and the motility score shows that the milt is viable for fertilization, and under 1 is not able to first fertilize properly. So this was measured over 49 days. As we see, the normal extender after 28 days reaches the 1 score, whilst the Ceretec extender keeps up in the 2 score. And after 35 days, the normal extender is not viable anymore, whilst the Ceretec stays viable for fertilization. And even after 49 days, we can see the score is just under 1. So this shows how the Ceretec can also give a possibility to improve the milt quality and the average motility score to this sperm in salmon. This is important because normally milt is preserved to wait for the ova in females. So you can't really wait that much long, and enabling a longer viability of the sperm can help the reproduction, in this case of salmon, trout, and other species. This is, I wanted to show these results because I understand, and Musa let us know that reproduction in ornamental is quite an issue, and some species is quite complicated. And maybe the fact that you can store the milt for more time using Ceretec can give a big help to those reproduction trials. So with this, I would like to conclude. With Ceretec, we hope to help the industry, both the ornamental and the aquaculture industry, at the same time that we improve welfare and condition of the fish. And I would like to thank the invitation and offer myself for any question during the Q&A section. And thank you very much for the attention.
Thank you. For Marta, your presentation is interesting talk and many information. Now is question and answer. We discuss about our talk today. I open session 1. I have many questions. Maybe I choose one question for each speaker. First, for Mr. Ahmad Musa, the question from Janu from Jambi. The question is like confirmation for Musa. Arona sect detection kit, is it mandatory or voluntary for arowana fish farmer? And how about other species of arowana, like S. formosus, etc.? Is it compatible for sect detection? Thank you. I choose only one. Actually, several questions. For Charles Saliba, the question is, I think it's general question. Whether every of Indonesian fish species need nutrition? Maybe the second question is, how far fish handling can effective stress, and what kind of genes that regulating heat shock protein? Question for Dr. Charles. For Marta, okay, why using nitrate when challenge test utilization of Ceretec in angelfish? Why not use another stressor agent? I think session 1 is enough. One question for each speaker. Let's see if we have enough time. We continue with session, second session. Okay, let's Pak Musa to answer the question.
Okay, thank you, Dr. Rafli. To Mr. Janu in Jambi, thanks for the question. Of course, it is not mandatory because we don't have the regulation until now. But what we suggest is, if the kit already established and we can practice in the field, so I guess even the farmer will agree with me that to protect our sovereignty in the ornamental fish, maybe only male fish should be export. Because what? In 2014, if I'm not mistaken, there is a news in Compass newspaper that Chinese already cultivate the Brazilian arowana, and we should be concerned of this. If they could cultivate the Brazilian arowana, what about Indonesian arowana? It is not possible if they have the stock, they have male and female fish. That's why I think the determination kit is very important for the field kit to avoid this condition. And about another species of arowana, I would like to give a screen share. Wait. This is the type of variant of arowana. There are Sclerophagus formosus, Macrocephalus, Giardini, Legendary, and Aureus. Actually, nowadays it only divided into two species, that's Formosus and Giardini. It's a debate on the scientist dialectic. What we find from these species that, did you see the screen? This, I would try to explain that we already purified and isolate several phytogenin from all of 4 variant of arowana. This is Banjar with B called Giardini with G and Pino, and then Super Red. What we found is the phytogenin of the 4 arowana. There is only one difference between another arowana. Is the specific one is the Giardini. It has 2 phytogenin type with this 180 and 120 kilodalton. This is the only differences. But usually when we measure or we make a cross reaction between this arowana phytogenin, the Giardini phytogenin will recognize another phytogenin. So if once we have the determination kit for arowana, it will compatible with another species of arowana. So don't worry that we will work twice. We only work once, and we believe that it will be fixed if we already have one phytogenin detection kit. Maybe I think it's all, and thanks for the question. We hope that we can protect our arowana fish, because it is really also harm for our farmer. Thank you. I give back to the moderator.
Thank you, Pak Musa. Let's we hear Dr. Charles Saliba to question for you. Time for yours.
Sorry, could you repeat the question, because it was very crackly, the line when you spoke to me. Could you repeat the question again?
Yeah, the general question, the first, whether every of fish, Indonesian fish species with nutrition.
Sorry, could you speak up? I can't hear you. Sorry, could you repeat the question? I can't hear you. Hello? Sorry, could you?
Just a moment, John. You hear?
Yes, I can hear you now.
Yeah.
Thank you.
Whether every of Indonesian fish species need nutrition is okay? Do you?
Whether nutrition is a stressor, is that the question?
Whether every fish species need nutrition. I think general question.
If I understand the question, your question is nutrition a stress? Is that the question?
Not specific stress, only general question. Whether every of fish species need nutrition.
Nutrition is one of the major factors of stress. Animals in general are either carnivores or omnivores. When we eat flesh, for example, there are many other elements to a feed other than just protein, carbohydrates, fat, ash, etc. The microelements, the micronutrients, and we see this a lot in our work, are essential. And very often we don't even know what those nutrients are and their role or their importance. So whenever anybody in any species is formulating a diet, technically a diet is an artificial diet. We are not feeding fish, are generally fed on fish. They're not fed pellets. So when we are producing a pellet in the form of a feed, it's very, very difficult, if not impossible, to get exactly what the nutrient levels in a fish would be. So because these elements would be missing or lacking or not in the right proportion, the ability to convert them into energy and growth are very difficult. So yes, from our experience in all species, and we work a lot all over the globe with not just aquaculture, nutrition is one of the biggest problems, one of the biggest issues in farming and mainly associated with stress. To give you an example, perhaps closer to home, to Indonesia, tuna. We work very closely with tuna farmers in Malta. Malta is the biggest producer of tuna in the Mediterranean, the bluefin tuna. This is a very high value fish. Today, the limiting factor to the growth of the tuna industry is nutrition. Nobody has been able to come up with a diet that can be produced in a feed company to feed tuna. So consequently, we still have to feed fish, mackerel, squid, to tuna in order to be able to grow the tuna. So that gives you an indication how important nutrition is. It is very, very, very difficult to produce a diet that will mimic the actual feed that the fish would be eating itself. So yes, from our experience, one of the biggest stressors in life is feed, yes.
Okay, the second question is, what kind of genes that regulating heat shock protein?
There are a number of stress genes that are upregulating. And so the stress genes regulate, they're either upregulated or downregulated. And then there's a whole mechanism, there's a whole cascade. So there's a heat shock factor, which will stimulate the specific heat shock. So it isn't one gene, it's a whole cascade of genes which are either upregulated or downregulated following an oxidative stress. This is the new area of research today, the genomics of stress genes. It's a huge area that's being developed.
Okay, thank you.
Thank you for your questions.
For the Marta, why using nitrate when challenge test circulation of Ceratec in angelfish? Why not use another stressor agent? Marta, time for you.
Thank you. Thank you for the question. We've done several trials in Ornamental. This is just a small recap of the trials. We've conducted several in Ornamental and in aquaculture species, and low quality water can be represented through nitrite, ammonia, or contaminants, but nitrite can directly be related to the quality of the water because of contamination, of fertilization of the fields next to the ponds. So this can cause a high nitrite concentration in the ponds, leading to a low quality and obviously a high mortality of the fish. But I mean, low quality water depends on the fish you are evaluating, basically. And this is just an example of low quality water.
Okay, thank you, Marta.
You're welcome.
I think the committee confirmed information that it's enough only one session because I come 3.5 hours. It's very long time webinar this afternoon. Yeah, the committee informed that all participants who want to absentee, please click on the link we have put on the Zoom meeting and also in YouTube. Please do, because after the closing this webinar, the absent will be closed. Okay, for the closing, ladies and gentlemen, finally, we come to the end of the part of our presentation. We would like to say thank you again to presenters for the informative, interesting talks, and to the whole audience for the active participation and some questions. We are sorry, some questions we cannot read and we answer. Hopefully, the presentation will be beneficial for everybody. Again, thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh. The time for the next session return to MC. Please, Mbak Bunga.
Before that, I want to inform after this session, after we close the ceremony, maybe some participants still want to have a discussion. If Charles or Marta still have time, we open the free session after the closing ceremony. Do you still have time, Marta and Mas Charles? Okay. So we will open the microphone after this. But please, the master of ceremony to close this ceremony.
Okay, thank you to our moderator, Dr. Afri Johan. And this is the last, and I will close this seminar. Before I'll close the seminar, I would like to say thanks to all of the participants, and I would like to apologize if I made something mistake. And thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Maybe Pak Musa will lead.
Yes, I will mute on the mic, but please, if you don't want to, you can asking and.
The host would like you to.
We have Mbak Nina Meliza.
Oh, berarti? Berarti itu kakak diaktifkan suara kakak.
There are several questions about application of Ceratec in of audience asking about
Iya.
how to apply and if possible through the fee that
Saya kadang teman-teman ada mau
Coba
malu.
identifikasi.
Thank you, Honors.
they want to give a
Kita
question.
kan juga mau ngapakan kita ini ya, betul itu langsung.
Melisa, where is Nina Melisa?
I think it's better if they type the questions because I can't hear anything.
punya aku kuesioner yang mana tuh?
Some of
Sebenarnya banyak, ada yang
researcher
yang Linda,
didn't hear.
yang kuesioner
Okay,
buat
so I will
rumah, yang buat
mute
Jepang.
all the participants.
Yang biasa kan udah punya
I
dulu.
will mute. In the
Yes, better. If
discussion
not, you
in
can't hear the
private-
question.
private chat.
Or you can,
some-
Musa, you
Yes.
can allow the person that wants to ask, they can tell you and you can unmute one person, not all.
Yeah. Okay, who want to give a question, please raise hand. If you afraid to give a question in English, you may speak in Bahasa and then I believe Martha understand.
No way.
Oh, yeah. Okay.